So we all remember that infamous story, Green Eggs and Ham, from elementary school, right? Now, what if I told you the science behind making those green eggs and ham a reality is also super crucial to cutting edge technologies today? That's right, synthetic biology is making waves in ways we didn't even know was possible, and it's directly impacting healthcare innovations. Today, I'm going to show you how. So, what exactly are you looking at here? Right now, you're looking at a plasmid, which is a small circular DNA molecule. Plasmids, think of them like DNA delivery trucks. They're used to introduce new genes into cells. So, what I'm going to be talking to you today about is my process of editing the plasmid backbone PXP522 with the EGFP protein. So, basically, what that means is my plasmid will now glow green under UV or blue ray light. And this is super important in the synthetic biology and gene editing field as it helps researchers and scientists track and also notice where specific therapeutic genes are expressed and everything is working right. So, firstly, my entire process began by adding my TEF1 promoter. So, my TEF1 promoter right now is currently highlighted and it's in yellow. So, my TEF1 promoter is what indicates the start of transcribing the downstream gene, in this case, which would be the EGFP. So, to insert a TEF1 promoter and start this insertion, you need to find a restriction site. So, I use the restriction site for the enzyme SACI, which I visually located when looking at the gene, and I digested the ends to make a sticky little entryway for me to insert the TEF1 promoter seamlessly. So after adding my TEF1 promoter, my next step was to add the Kozak sequence. So you can see my Kozak sequence is currently highlighted and you can see it on the plasmid as well, slightly in the light green. It's super small, but a Kozak sequence is essentially just a short sequence of nucleotides, and they act as the initiation site where protein translation begins, specifically in eukaryotic mRNA transcription. It's super essential for helping the ribosomes recognize where they're supposed to start translating the mRNA into protein. So, now for my most major step, the actual insertion of the EGFP protein. So the EGFP protein was just copy pasted from an external database where I found the sequence for it, but let's do a quick little deep dive into how this works. So now that we have done our prep work, which is the digestion of the cus site, our Kozak sequence, and our TEF1 promoter, the EGFP protein can now be added, and all the previous work will allow it to be transcribed and translated correctly. So, the EGFP protein is essentially just shaped like a barrel, and it's a modified version of the GFP extracted from a jellyfish. So, inside this barrel, there is a chromophore, and this is the part of the protein responsible for creating the green light. When matured, these acids in the chromophore emit a bright green light when it's exposed to UV light or blue light, and which is super important for gene tracking. So similar how we had to take pre-process and steps before adding the actual protein, after inserting the protein, there are also steps we need to take after it to ensure that it is translated and transcribed properly. So, right after inserting the protein, the next thing I did was add my stop codon. So, you can see my stop codon right now highlighted in orange. And a stop codon is a sequence of three nucleotides that signal the end of protein synthesis. So, while it is only three nucleotides, it is still super important that it makes sure that the protein is made to the right length without other unwanted parts which can mess with the function and the overall function of the plasmid as well. And lastly, my final step in this entire editing process was to add the terminator. So a terminator sequence that I used was CYCI, and the terminator sequence, similar to what the name suggests, as well as the purpose of the TEF1 promoter in the beginning, is the final step that counteracts the initial TEF1 promoter. So the terminator sequence, instead of signaling the start of a transcription, it signals the end of the transcription, making sure that the other regions of the plasmid are not interfered with or disrupted by the gene, and that the transcription will end precisely, making sure that the protein transcription has happened in the area it needs to and has happened and completed successfully. So, while I briefly mentioned this earlier, the opportunities of implementation for this newly edited plasmid are truly vast. And today, I'm just going to talk you through some of the key ways that this plasmid can be implemented in the healthcare sector. So, let's dive right in. First up, we have CAR T cell therapy. CAR T cell therapy is a cutting edge treatment where a patient's T cells, those powerful natural defenders of your body, are edited so they can specifically target and destroy cancer cells. By adding this plasmid to modified T cells, scientists and healthcare professionals would be able to track them in real time, allowing them to ensure that the therapy is working as intended, as well as allowing them to make any changes as needed to optimize the treatment. This precision can be a huge game changer for the battle against cancer. Next, let's talk about gene editing. So, this is an area of medicine where defective genes in a patient are either corrected or replaced. And when you're making such precise edits to the genome, it's super important to make sure everything is done precisely and correctly to minimize the risk of side effects. So, this is where the GFP protein would come into place. With this fluorescence marker, scientists and researchers could accurately and visually confirm that any edits made have been precise and controlled, and minimize the risk of unintended effects by ensuring that any edits have successfully been made. It's an essential tool for advancing gene therapy to make it a lot safer for all. Finally, let's look at drug modeling and development. So, one of the biggest challenges in the drug development industry is understanding how does this drug react with the target disease. And researchers need to be able to observe the behaviors, outcomes, as well as the interactions of the drug in real-world time to truly be able to understand how effective the drug is. So, by using this newly edited plasmid to tag specific target genes in the disease, researchers would be able to actually visualize and gain deeper insights of how the drug is being used and where improvements need to be made and how effective the drug is being as well. So, this not only accelerates the drug screening process, but it really helps with the drug discovery process as well, bringing new therapies and new drugs to patients even more faster and more efficiently. So, to recap, this was my process of how I edited a plasmid, as well as its various applications in the healthcare sector. It's crazy to think something as small as a little glowing protein, combined with the power of synthetic biology, can create something so revolutionizing to the healthcare field and drive such innovative research. From stuff like drug development to creating safer therapies, the field of healthcare is vast and the improvements and advancements are crazy as well, and we've only just scratched the surface. If you want a more detailed version of my experiment and its applications, I'll leave a link to a full article in the description, and thank you so much for watching.